Hello there and welcome to Just About Earth, where curiosity finds a quiet corner and science slips off its shoes for a while. Tonight we're venturing into a realm so old, so strange, it predates flowers, forests, and even the idea of soil. A realm where the first pioneers of decay and transformation quietly reshaped the very skin of the planet. Tonight we're asking, what was the first fungus like? It's a gentle mystery, wrapped in threads finer than hair, rooted not in grandeur but in rot, which, in its own earthy way, is quite beautiful. Fungus is nature's recycler, its silent architect, its patient thread running through time. But before it was the quiet force we know today, sprouting mushrooms after rain or turning old bread into fuzzy science experiments, it began as something far humbler and far older. So let me ask, where are you listening from tonight? And when you close your eyes, can you picture something alive, yet without a face, a brain, or even a true body? Something neither plant nor animal, but older than both. If you find this quiet corner comforting, if the idea of wandering your way through deep time soothes your evening, a like or a subscribe would be ever so kind. It helps this little channel find other sleepy seekers wherever they may be in the world or in the timeline. Now I do hope this doesn't make you so fascinated that you lie awake imagining spores and ancient woodlands, but if it does, well, at least you'll have gained a new appreciation for the secret lives beneath your feet. So settle in, slow your thoughts, and let's begin. Because over 800 million years ago, long before leaves rustled or insects buzzed, something began creeping across the bare rock of Earth, not with legs or roots, but with filaments. It didn't roar, it didn't bloom, it spread. And from that quiet beginning, life would never be the same. In the vast silence of Canada's Northwest Territories, just shy of the Arctic Circle, there lies a place called the Mackenzie Mountains. A quiet, weathered land where time wears thick boots and speaks in stone. Among the shale and silt of these lonely outcrops, scientists noticed something peculiar. It wasn't dramatic, not at first glance, just a slice of rock, thin as a cracker, flecked with ancient sediment. But when examined under a microscope, it revealed something the Earth had been holding onto for nearly 1 billion years. Filaments, branching, delicate, almost like a web caught mid-spin, and at the tips of some of those threads, tiny bulb-like swellings, no bones, no shells, no obvious signs of movement or purpose, but unmistakably alive once, and unmistakably fungal. That rock came from a place called the Grenville province, and it dates back around 890 million years. That's older than the first animal, older than the first forest, older, even, than the continents in their modern positions. And it may be our very first fossil record of a fungus. The find was made by a geologist named Corinne Tremblay, part of a team who had been studying sedimentary layers laid down when this land was part of an ancient seabed. They weren't looking for fungi. They were tracing tectonics, following the signature of an ancient mountain-building event. But science, like fungus, often spreads where you least expect it. To confirm the fossil's identity, the sample was sent to a paleobiologist, then to another, and another. Each layer of analysis peeled away a bit more doubt. What they saw were chitin walls, the tough, flexible material that still makes up the cell walls of modern fungi, and those branching patterns, so precise, so eerily familiar. Not random mineral formations, not bacterial residue, something more. And just like that, a quiet revolution stirred in the timeline of life. Until that discovery, the earliest confirmed fungi were thought to be around 500 million years old, just slightly older than the first plants. This pushed their origin back by almost 400 million years. And not just back in time, but into a very different world. To understand why that matters, we need to step backward ourselves. Not just hundreds of years, or thousands, but nearly 1 billion. We find ourselves in the middle of the Proterozoic eon, a time before anything had yet bloomed, buzzed, or breathed in the way we think of today. The air was thin. Oxygen was only beginning to make its slow climb into the atmosphere. The continents were lumped together in awkward configurations, their coastlines etched by tides but barren of green. There were no birds, no grass, not even moss. And yet, beneath the shallow seas, life was stirring. Slow, simple, but persistent. Microbial mats clung to the seafloor. Single-celled organisms drifted in sunlit shallows, practicing photosynthesis like a slow cosmic prayer. Stromatolites, domes of bacterial colonies, were already ancient by this point, having sculpted their way through eons. Life was still largely microscopic, and the land above sea level was a hushed expanse of rock, wind, and silence. So where, in this quiet world, would fungus begin? Not with mushrooms, not yet. The fungus we know today, morel, shiitake, fairy ring, are the fruiting bodies of vast underground networks, mycelium. That's the real body of a fungus, and it's likely that the earliest forms were little more than branching threads, hyphae, stretching across whetstone or decaying organic matter, if any existed. We imagine a coastline, low, flat, without trees. The tide retreats. The rock glistens, warm from a sun that burns slightly dimmer than today's. And there, so fine you'd miss it, grows a patchwork of hyphal strands. They spread slowly, anchoring to mineral surfaces, secreting acids to extract nutrients. Not feeding in the traditional sense, but digesting outwardly, breaking down whatever lay beneath. A kind of chemistry without eyes or hunger, just function. This is the beginning of a concept, the external stomach, a trait still unique to fungi. Instead of taking food in, fungi dissolve the world outside themselves and absorb it. They don't consume, they merge, and that may have been their first great power. In an ecosystem where very little could break down rock, early fungi might have been one of the first forms of life capable of truly changing the surface of the land. Their secretions could weather stone, releasing minerals. Their threads could trap and hold water. In doing so, they may have prepared the ground, quite literally, for the future arrival of plants. In fact, some scientists believe that fungi made plant life on land possible. Today, over 90% of plant species form partnerships with fungi through structures called mycorrhizae. These fungi wrap around or even grow into plant roots, exchanging minerals and water for sugars. It's one of the oldest and most successful biological alliances on Earth. But in the Proterozoic there were no plants yet. Only fungi, perhaps slowly carving out niches in the cracks of rock, feeding on microbial mats, or collaborating with early algae in lichen-like forms. Small, silent, unnoticed. They were the backstage crew of life. The soil before there was soil. And yet, their chemistry would begin altering the planet. By breaking down rock, fungi contributed to the formation of soil, an invention so fundamental we rarely think of it as one. No soil, no plants, no plants, no animals, no animals, no us. The fossil from the Mackenzie Mountains didn't just push back a date, it whispered of an ancient inheritance, one we all share every time we walk on ground softened by decay. And here's the quiet twist. Fungi aren't plants. They're more closely related to animals. It's an odd kinship stitched deep into the genome. Both fungi and animals share a common ancestor, likely a unicellular creature with a flagellum, a whip-like tail for swimming. It's possible that the early ancestors of fungi swam too, living in water before ever touching land. And then, one day, or rather over millions of days, a lineage began creeping onto rocks, clinging, growing, changing. And over time, they became the first terrestrial colonists. Millions of years later, when plants made their debut on land, fungi were already there, waiting. Some likely welcomed the partnership, others continued the quiet work of decay, of transformation. They helped shape the carbon cycle. They helped build ecosystems. And during the most dramatic events in Earth's history, the mass extinctions, the volcanic winters, the asteroid aftermaths, they were among the survivors. Because fungi don't need sunlight. They thrive in darkness, in damp, in rot, and when life above crumbles, they bloom below. Perhaps that's why they've always felt a little magical, a little otherworldly. They appear overnight, they vanish by noon, they glow in the dark, mimic textures, and produce compounds that can heal or harm. In ancient cultures, they were linked to spirits, to death, to dreams. And in modern science, they're emerging as key players in everything from climate resilience to neurological research. But it all began, as so many things do, not with a bang, but with a branching thread, with a tangle of hyphae spreading across stone, unhurried, uncelebrated, unnoticed. Until now. Today, scientists continue to uncover ancient fungal traces, biomarkers, microfossils, chemical signatures trapped in billion-year-old stone. They debate, they analyse, they imagine, and in that imagination, we begin to glimpse the outlines of something astonishing. Not just what life is, but how it began to prepare the world for everything that followed. So when we ask, what was the first fungus like? The answer is not a name or a picture, it's a feeling. Quiet, patient, indispensable. Not quite a creature, not quite a concept, but a soft, persistent presence that has always worked in the margins. Even now, under your feet, in the dark folds of forest loam, or the damp cracks in a city sidewalk, fungus is still telling the story of life. Not the glamorous chapters, but the ones that hold everything together. The glue, the rot, the beginning, and it started here, in stone, in silence, in strands. To understand the first fungus, we have to forget everything we know about life on land. No forests, no birds, no insects humming through open petals. The very air, thin and raw, held only a fraction of the oxygen we breathe today. The sky was likely tinged with a paler blue, and the sun, unfiltered by a thick ozone, poured down with a steady, unrelenting glare. This was a different Earth. Let's go back, somewhere between 900 million and a billion years ago. The continents were little more than smudges of ancient rock, low-slung and scattered across a planet still learning how to arrange itself. What land there was sat bare, windswept, and silent. No roots held the soil, no branches caught the breeze, and that's because, quite simply, there were no plants, not yet, not even moss. Instead, the world was shaped by water and stone. Seas stretched shallow and wide, spreading across low-lying coastlines. Tides were longer, pulled harder by a moon closer than the one we know now. Beneath the waves, the base of the food chain was microbial, cyanobacteria floating in sunlit currents, forming sticky colonies on the seabed. Stromatolites, ancient microbial mounds, grew slowly in the shallows like rock sculptures built grain by grain. The ocean itself was a soup of chemistry, rich in iron, poor in oxygen. Methane bubbled from hydrothermal vents. Volcanic gases vented from rift zones, lacing the atmosphere with sulfur and carbon dioxide. Lightning flickered over warm, turbulent water, and the continents, such as they were, creaked and drifted, still assembling the foundations of familiar geography. But on the margins, where land met sea, where the tide withdrew and left a rock to dry, something subtle was beginning. The land was not dead, exactly. It was waiting. And life, it turns out, is always looking for a foothold. That first fungus, whatever it was, likely didn't announce itself. It didn't rise like a tree or shine like a bloom. It crept. Its body, a mycelium of hyphae, thin and branching, spread quietly across damp rock. No leaves, no chlorophyll, just a network, a living latticework, reaching outward in every direction, mapping cracks, secreting enzymes, dissolving whatever it touched. Fungi don't eat in the usual sense. They digest externally, releasing acids that break down complex molecules into simpler ones, sugars, minerals, even the bedrock beneath them, slowly pried apart by chemical fingers. In those early eons, there was very little for a fungus to feed on. No fallen leaves, no dead trees. But there were microbial mats, dense carpets of photosynthetic bacteria clinging to tidal flats and submerged surfaces. Perhaps the earliest fungi fed on those, parasitizing or decomposing their biofilms. Or maybe they formed symbiotic relationships, coexisting, sharing nutrients in exchange for structure. Another possibility, equally plausible, is that early fungi lived in partnership with algae, together forming lichen. Today, lichens colonize some of the most hostile places on Earth. Bare rock, mountaintops, desert crusts. They're early settlers, slow, patient builders of ecosystems, and some researchers believe that this ancient alliance may date all the way back to the birth of terrestrial life itself. Imagine a thin green-grey crust spreading over volcanic rock, catching mist from the air, holding it just long enough to create a micro-environment. Below, the hyphae stretch, anchoring the life form. Above, the algae photosynthesize. Together they survive where nothing else can. This would have been the first soil, if you could call it that. Not the loamy, root-packed earth we know today, but a fragile living film, an interface between geology and biology, a beginning. And like all beginnings, it was slow. For millions of years, the land changed little. But change it did. As fungi and microbial life crept further inland, they began to alter the chemistry of the rocks they lived on. Their acids broke down minerals, releasing elements like calcium, potassium, and phosphorus, nutrients vital for future life. They captured carbon, releasing organic acids in return. Slowly, imperceptibly, the land became more hospitable. Fungi, though they lack ambition in the human sense, are tireless. Their networks don't sleep. They explore. They respond. A single mycelial network can stretch for miles, communicating chemically, transferring signals and nutrients in patterns we're only beginning to understand. And even in those earliest forms, those billion-year-old ancestors, it's likely that similar principles were already in play. No thoughts, no nerves, but a kind of distributed intelligence. This is not the mind we're used to, but it is a form of knowing. And so the land was not just being colonized, it was being understood in a fungal way. Mapped, metabolized, made ready. Over time, these networks likely grew in complexity. They adapted to different conditions, dryness, acidity, cold, and in the process, they laid down a foundation that would prove invaluable for what came next, the greening of the earth. But let's pause here. Let's not rush into forests and ferns just yet, because there's something uniquely beautiful about this chapter. This pre-plant world, this era where the only life on land wore no green, bore no fruit, cast no seeds. It simply spread. And in that spreading, it sculpted. We often think of life as something visible, colourful, moving, dramatic. But fungi challenge that instinct. They are understated, cryptic. Most of their mass lies underground or out of sight. What we call mushrooms are only the briefest flickers of their presence, the reproductive structures, the fruiting bodies, the temporary flare of something vast and hidden. That concept, that most of the life is invisible, was as true 1 billion years ago as it is today. Fungi were, and remain, the backstage crew of evolution. They manage decay, they build soil, they unlock nutrients, and in doing so, they support everything else. This principle holds across deep time. When the first plants eventually did appear on land, simple liverwort-like forms growing close to the ground, they were, in many ways, helpless. Their roots were shallow, their ability to extract nutrients was poor, but they partnered with fungi. Mycorrhizal networks formed around their roots, exchanging minerals for sugars, And this partnership transformed the biosphere. Trees rose, forests followed, the atmosphere changed, and beneath it all, fungi kept growing. They helped life take hold, and when life died, they helped return it to the cycle. But long before the drama of trees, long before animals took their first steps, even before multicellular life made its debut in any meaningful form, fungi were here. Their early ancestors may have looked simple, just threads, just tubes. But those tubes carried enzymes, and those enzymes carried possibility. And when we find traces of those ancient threads, preserved in rock, sealed in shale, brushed by hands in sterile laboratories, we're not just seeing fossils, we're seeing the prelude. Because every era of life builds on the one before it, the future buried in the past, the fruiting body born of the mycelium, the oak dependent on the rot of its predecessors, and the story of Earth, the true, full story, includes not just the stars of the stage, but the quiet ones in the wings. Fungi remind us that transformation doesn't always announce itself. Sometimes it begins with a spore, invisible, drifting, landing on a surface that no one else wanted. Sometimes it spreads in darkness, following moisture, sensing minerals. Sometimes it waits for years, decades, eons, until the conditions are right. And then, quietly, without permission or applause, it changes the world. That's what the ancient Earth looked like. Not a garden, not a jungle, not a canvas already painted, but a canvas just beginning to stretch. And fungi, they were the first brushstrokes. There is a pattern in Earth's story, a heartbeat of creation and collapse, emergence and extinction. And nestled deep within those rhythms is a truth as old as life itself. Change rarely comes with ceremony. It begins as a whisper, a shift in temperature, a tremble beneath the crust, a molecule misplaced. And sometimes change comes not in the form of disaster, but in the form of opportunity. And sometimes the mechanism that shapes a world is not a bang or a blaze, but a slow bloom of chemistry. The earliest fungi evolved during one of those long silences in Earth's history. Not a catastrophe, not a cataclysm, a quiet era, the late Proterozoic, a time of continental restlessness and biological murmurings. Continents were still wandering, the atmosphere was still finding its balance, and life, which had for billions of years been the sole property of oceans, was starting, timidly, to peer above the waves. But the land wasn't welcoming. Not yet. Imagine standing on a Proterozoic shore. The wind is harsher, the sky more violet, tinged by an atmosphere still short on ozone. The ground beneath your feet is rough, raw, made of unweathered basalt and granite. There's no soil, only stone. The waves are thicker somehow, richer with minerals, foamy with microbial residue. And everywhere is silence. Into this, something small begins to stir. Not in rebellion, not in conquest, just in search of space. Fungi, these earliest colonists, didn't arrive all at once, they crept, and their arrival set in motion a chain of chemical events that would change the planet's surface forever. It began with rock. Our planet's crust is made of minerals, locked, inert, hard. But life, even the earliest microbial kind, has a way of unlocking what was never meant to move. Fungi do this by excreting organic acids, oxalic acid, citric acid, and others. These acids break down silicate minerals, freeing up elements like iron, magnesium, and phosphorus. Phosphorus, in particular, is vital. It's the backbone of DNA, the scaffolding of cells. Without it, life cannot grow. Early fungi were, in effect, biogeochemical alchemists, transforming stone into nutrient. And this weathering didn't just affect the rock, it influenced the atmosphere. As fungi broke down minerals, they facilitated carbon capture. The release of calcium and magnesium helped trap carbon dioxide in carbonate rocks, pulling greenhouse gases from the air and lowering global temperatures. Slowly, subtly but persistently. Over millions of years, this contributed to one of the Earth's great cooling periods, a planetary ice age so deep and so thorough it may have frozen the oceans all the way to the equator. Scientists call it Snowball Earth. Was fungus the cause? Not entirely. But it may have been a player, one among many, a quiet accomplice to a global transformation. Imagine it. Continents dusted with frost, glaciers grinding across tropical belts, sunlight reflecting off ice, reinforcing the cold, oceans sealed beneath thick sheets, and yet beneath it all, in the fractures of rock, in the brine pockets and crevices, life endured. Fungi are resilient. They thrive in darkness, in cold, in acidity. Modern fungi have been found in Antarctic permafrost, inside nuclear reactors, deep in caves sealed for millennia. If any organisms could survive a global deep freeze, it would be them. And survive they did. When the planet finally began to thaw, driven by volcanic outgassing and the slow build-up of CO2, life emerged again, blinking into the warmth. And the land, now dusted with the remnants of fungal weathering, was ready for a new chapter. This was the trigger. Not a single event, but a prolonged series of interwoven changes. Tectonics pushed continents upward. Erosion exposed fresh rock. Fungi crept across it. Chemical weathering pulled CO2 from the sky. The planet cooled. Ice blanketed the world, and beneath the ice, life evolved. Fungi, by dissolving rock and preparing soil, had quietly changed the rules, and they weren't done. As ice melted and shorelines expanded, new environments opened up. Tidal flats, deltas, moist, mineral-rich zones where hyphae could stretch farther and deeper. And where fungi spread, they carried with them an ability that was about to reshape evolution itself. Symbiosis. Somewhere in this post-glacial world, perhaps among the cracks of a newly thawed cliff, fungi encountered photosynthetic algae, or maybe cyanobacteria, ancient masters of sun harvesting, and instead of competing, they combined. This wasn't a conscious decision. It was chemistry, survival, a merger of strengths. The fungus provided structure, moisture retention, and access to minerals. The algae provided sugar, energy from light. This was the birth of lichens. And lichens, as it turns out, are more than just crusty growths on rocks. They're ecosystem engineers. They colonize bare surfaces. They trap dust and water. They create microhabitats, and they accelerate weathering even further. Once lichens appeared, the pace of terrestrial transformation quickened. More rock was broken down, more nutrients released, more carbon captured. Soil, in its earliest form, began to accumulate. Thin, fragile, but real. And this soil was the cradle in which future ecosystems would take root. So in this chapter of Earth Story, the mechanism of change wasn't explosive. It was filamentous. It was chemical. It was slow. Fungi didn't reshape the world with force. They did it with persistence. They did it by showing up where nothing else could live. They did it by breaking down the unbreakable. And in doing so, they made room for everything else. When we talk about evolutionary breakthroughs, we often picture bold forms, the first backbone, the first eye, the first flight. But fungi remind us that some of the most profound revolutions begin not with invention, but with decomposition. The ability to dismantle, to recycle, to turn the dead into nourishment, to make endings fertile again. This, perhaps, was the first great trick of complex life, and it belonged not to predators, not to builders, not even to thinkers, but to decomposers, to fungi. The world that thawed from Snowball Earth was both familiar and utterly alien. A stripped clean canvas shivering beneath a pale sun, its colours sharp and new after millennia locked beneath ice. Glaciers carved into emerald seas whose surfaces steamed under the sudden change, and torrents roared down raw granite slopes, carving braided rivers that glittered like loose threads of silver embroidery. Each flood ripped fresh mineral from the continents, sluicing iron and silica and trace metals into shallow basins where life, though shaken, never truly slept. There, in brackish deltas and tide-pulsed flats, fungi met the moment with the patience of stonecutters and the hunger of chemists, spinning their hyphae through mud the way constellations trace darkness, turning dissolution into design. Their acids etched feldspar into clay, freed phosphorus loose from apatite, coaxed potassium and calcium from their crystalline shelters, and every liberated ion became a note in an ever-growing biochemical chorus. Microbes drifted through the soup these elements thickened, blooming in sunlit shallows, fixing carbon in frantic photosynthetic pulses. As they died, their bodies rained onto the seafloor in snowdrifts of organic matter, and fungi, already masters of the rocky shoreline, followed, infiltrating mats of decaying cells, inventing the art of soft tissue recycling long before scavengers dreamed of teeth. Each filament released a sigh of carbon back to water, but every atom fixed sturdily into carbonate rock or buried beneath silt was one less warming the air above. The climate wavered between chill and thaw like a pendulum seeking its cadence, and fungi were the invisible fingers nudging it toward balance. Above the tide line, lichens colonized cliffs in a slow explosion of ochres and sage greens, stitching moisture to mineral in the world's first terrestrial tapestries. Frost needles wedged into cracks they'd started, rain-washed microscopic gravel into hollows where dark biofilms waited to pounce on nutrients, and grain by patient grain a proto-soil thickened. It was not the dark loam gardener's prize today, more a transient smear of dust held together by slime, but it trapped seeds of possibility. When volcanic islands belched fresh basalt into warm oceans, spores hitchhiked on wind currents to land on infant coastlines still hot from magma. There they cooled the stone, netted salts, and once again began the long arithmetic of turning black rock to brown earth. Every ridge they softened became a future refuge for pioneering plants. Every fracture they widened became a corridor for water. Every atom of phosphorus they mobilized primed the planet for metabolic fireworks still millions of years away. Yet the mechanism of change was not merely chemical, it was networked. Within the mycelial labyrinths, pulses of electrical potential flickered, primitive whispers of information relayed through cytoplasmic highways. When one hyphal tip encountered a cache of iron or manganese, chemical messages surged backward, rerouting growth, reallocating enzymes, synchronizing digestion along kilometers of living thread. The planet's first broadband, invisible and silent, was already online under that barren crust, coordinating weathering on a continental scale. Feedback loops emerged. Richer soils fostered thicker mats of algae at the water's edge. Dead algae fed thicker veils of fungi. Thicker veils released more acids deepened the soil. The system tangled, tightened, reinforced itself, until coastlines once austere became mottled swaths of microbial colour, each patch an engine humming with exchange. Inland, katabatic winds swept across glacial outwash plains, lofting dust high into the atmosphere where it drifted for weeks before snowing down on distant seas. Iron-rich particles fertilized oceans starved after eons under ice, and algal blooms the size of small countries unfurled overnight. Their photosynthetic fury vented oxygen, nudging atmospheric levels ever higher, and though animals still slumbered in evolutionary infancy, that oxygen seeped into shallow sediments where fungi and sulfate-eating bacteria conducted secret alchemies, forging the planet's earliest complex sulfur cycles. Trace metals precipitated, banded, recrystallized. Ores we mine today for copper and zinc began as byproducts of fungal chemistry, scribbled across primordial shorelines. All the while, Earth's crust creaked, supercontinents rifted, sutured, rifted again. Every mountain-building thrust exhumed fresh, unweathered rock, and fungi marched in tandem with the peaks, colonising scree even as summits still rose. On talus slopes perpetually scraped by landslides, hyphae probed pores between quartz grains, weaving grey crusts that anchored pebbles, long enough for frost and heat to pry them finer. When rivers carried that sediment to floodplains, fungi were already present in the slurry, ready to knit silt into the thin, sticky veneers where the earliest embryonic plants, those liverwort-like sprigs no taller than a thumbnail, would someday take hold. The first of those plants likely emerged along equatorial marsh flats, frail green fronds clutching at damp soil, each cell wall trembling with the challenge of desiccation. Their rooting organs were mere rhizoids, filaments incapable of serious excavation, but fungal partners wrapped them in a living glove. Mycorrhizal hyphae plunged deeper than any plant cell dared, scavenging phosphate, buffering toxins, ferrying water molecule by molecule into plant tissue, and shuttling back the sugars that plants coaxed from sunlight. This barter arrangement, carbon for minerals, was evolution's masterstroke. It turbocharged both lineages, accelerating photosynthesis on land and accelerating rock weathering underground, doubling down on the drawdown of atmospheric CO2. Greenhouse gases waned, climates cooled again, and ice sheets advanced. Yet each icy episode arrived to a world incrementally greener than before, and thus retreated sooner, surrendered faster, because green means dark, and dark means heat absorbed. Through these cycles of freeze and flourish, fungi remained sovereign recyclers. When pioneer plants succumbed to drought or frost, mycelia liquefied their fragile bodies, releasing nutrients for the next generation of sprouts. When volcanic ash blanketed river deltas, fungal spores sprouted atop the sterile grit, initiating soil formation anew. And when lightning split the sky and birthed wildfires, yes, even in landscapes with no trees, lightning still found mats of desiccated vegetation to ignite. Fungi were first on the scene afterward, feasting on char, unlocking minerals bound in blackened biomass, sowing the ground with the promise of regrowth. Millions of years blurred into quarter-billion-year spans, and tectonic forces raised a supercontinent paleontologist now called Rodinia. Its interior baked under monsoons, yet its coasts teemed with lagoons rich in dissolved silica from relentless fungal weathering. In those warm shallows, protosponges and jelly-like animals experimented with multicellularity, threading collagenous tissues through calcium-infused seawater. Some researchers speculate that fungal activity on land was key to maintaining just the right balance of ions that allowed these early animals to form simple skeletons, for without sustained runoff of calcium and bicarbonate, their budding shells would have been chemically impossible. Thus, in a feedback intricate as lace, terrestrial fungus underpinned marine animal innovation, proving again that the true mechanism of change often hides in exchanges too small for ancient eyes to see. And still, the atmosphere altered. Oxygen, once scarce, crept above 1%, then 2, then 6. With each increment, ultraviolet light thinned, granting fungi and their photosynthetic partners freedom to venture farther from water's edge. Hyphae ventured into cave systems opened by rivers, exhaling carbon dioxide that mixed with groundwater to carve stalactites and stalagmites in slow-motion stonework. Surface networks infiltrated wind-blown dunes, stabilizing them, turning seas of shifting sand into embryonic grasslands where none yet existed, but where evolutionary potential gathered like storm clouds. The genetic story, teased from molecular clocks in modern fungal lineages, suggests bursts of diversification that match neatly with these environmental swings. Whole clades split when Rodinia fractured, each continental shard carrying its own suite of spores into isolation, free to adapt to local soils, local chemistries, local climates. Some lineages refined the craft of cellulose digestion, setting the stage for future forests. Others evolved enzymes to break lignin, an ability that billions of years later would determine the colour of coal and the global cycle of carbon through wood. Still others, faced with periodic inundation, learned to sporulate underwater, pioneering amphibious strategies later mirrored by amphibians themselves. In laboratory freezers today sit genes descended from those innovations, genes we borrow to brew beer, to bake bread, to craft antibiotics, to engineer biofuels. Their first drafts were penned in the Proterozoic dusk when no creature yet possessed eyes to watch. A billion years later, they are still scripting the possibilities of chemistry, reminding us that biotechnology is not a 21st century invention, but an inheritance from a planet that let fungus run its course. And what of the mushrooms, the visible eruptions we now associate with autumn forests and fairy stories? Their ancestors likely appeared late in this ancient saga, after plants were firmly established and woody debris littered the ground. But their precursors, the filamentous mats, the modest sporangia, the stealthy launch pads for spores, were already honing the physics of dispersal. Any breeze whispering across a damp stone could pick up 1 billion spores and loft them halfway around the fresh-cooled globe. Some would freeze, some would drown, but a handful would land on newly uplifted crust or freshly cooled lava, and from each survivor, the cycle of chemical conquest would recommence. Acids out, minerals in, rock undone, soil born. By the close of the Proterozoic, Earth's surface had been fundamentally rewritten. What began as barren basalt was now stippled with microbial mats, crustose lichens, tufted clumps of early land plants, and below them all, vast unseen webs of fungi. Rivers ran browner, loaded with organics that tinted estuaries like weak tea. Sediments layered thicker on continental shelves, rich in carbon that future tectonics would bake into black shales brimful of promise for curious primates yet to evolve. If you could hover above that world while holding a modern colour palette, you would see greys and blacks giving way, in patches, to olives and russets and faint, hopeful streaks of green. You would sense the atmosphere's flavour shifting, less acrid, more breathable. You would note the temperatures stabilising as weathering and photosynthesis found their uneasy equilibrium. You might even hear the faintest crackle of evolutionary static, proto-insects rustling among liverwort fronds, filamentous predators traipsing through mud, waves murmuring against reefs built not by corals but by bacterial thickets, testing skeletal complexity. And in every one of those scenes, fungus is there but seldom seen, orchestrating biochemistry with the discretion of a stagehand drawing curtains between acts. Its influence is planetary, yet its body is frugal, a wool 1 cell thick, cytoplasm flowing like sap, nuclei drifting in slow currents, genomes poised to rewrite themselves when opportunity or adversity knocks. That elegance, minimal structure, maximal impact may be the purest distillation of evolutionary success. It is a strategy older than wood, older than blood, older, in fact, than the very concept of root or leaf or bone. It is the strategy that allowed Earth's crust to tiptoe from sterility toward verdure. And so, when we speak of a trigger, we mean this slow, inexorable tipping of feedback loops set in motion by fungal chemistry. No singular asteroid, no single plume of lava, but a billion-year negotiation between rock and life, mediated by filaments that never once paused to wonder at their own significance. Without them, perhaps the planet would still be a waterworld flecked with stone islands too raw for green. Without them, perhaps oxygen would have stalled below levels fit for breath. Without them, perhaps carbon would have lingered in the sky, trapping heat until oceans boiled. Instead, they dissolved the obstacles grain by grain, clearing a path for complexity to stroll ashore. And when complexity finally did, when spore-bearing ferns sprung waist-high and dragonflies the size of kites buzzed over canopy, fungi continued their quiet vocation. They decomposed logs into humus. They partnered with rootlets to feed towering trees. They formed subterranean networks, wood-wide webs, that whispered chemical gossip from grove to grove. They healed, they hindered, they hallucinated, they haunted, they humbled. But all of those later glories rest on the foundation they built in those first unfathomably distant dawns, when the planet was newborn and the only certainty was stone waiting to be undone. That, then, is the long mechanism of change. Filament by filament, molecule by molecule, climate and crust reshaped by appetites too small to see, too faithful to stop, too fundamental to ignore. In the story of the first fungus, there is no climax, only accumulation. No hero, only habit. No miracle, only chemistry, repeated until the improbable becomes inevitable. And if ever you doubt the power of the patient and the unseen, step into a quiet wood, kneel, brush aside the leaves, and touch the fine white threads that bind every crumb of earth beneath your fingers. They are the living memory of 1 billion years of transformation, still at work, still humming their slow spell of dissolution and creation, whispering the same invitation they offered the first green shoot that dared lift its head to sky. Grow here. I will make the world ready for you. When a fungus changes a rock, the world does not shudder. There is no thunderclap, no crack in the sky, no instant flash of transformation. But give it time, give it a few thousand years, or a few million, and the world begins to tilt in quiet acknowledgement. Because in that moment when a hypha first threads its way into a crystal lattice and begins to break it apart, something subtle but irreversible happens. The land becomes permeable to life, and life, as it tends to do, rushes in. So what were the chain reactions of that first fungal wave, the ripples from that earlier spreading veil? We begin, again, with soil. Before fungus, the land was stone, silent, brittle. Water ran over it like oil on glass, unable to soak, unable to stay. Nutrients, even if released from the rock by weather or lightning, would vanish downstream, ephemeral, wasted. But fungus changed that, by carving pathways through mineral grain, by softening rock with its chemistry, by mingling its biomass with microbial mats and wind-blown dust. It formed something that held together, something that stayed. Soil was born, not all at once, but in microbial smudges and lichen-stained hollows, and with soil came permanence. For the first time nutrients could linger. For the first time water could be held just long enough to make growth sustainable on land. And then came the guests. Primitive plants, fragile liverwort-like beings with no roots, no stems, no vascular systems, barely more than green flaps, drifted in on spores or crept from shallows with halting confidence. And they found a foothold in fungal-prepared ground. But these plants could not have lasted long on their own. Their primitive rhizoids could not dig. Their biology could not draw in minerals from the stubborn rock. They were unprepared. And yet, they thrived. Because below them, wrapped around their simple rootlets, were fungal partners. The mycorrhizal handshake had begun. This was one of the most profound partnerships in the history of life. Fungus and plant, mineral and light, decay and growth in silent exchange. The fungus extended the plant's reach, acting as a mineral mining network, a water sponge, a protective sheath. And in return, the plant offered sugar, crafted in the sunlit cells above. This exchange didn't just help the plant survive, it accelerated its success. It allowed greenery to march inward from the coastlines, onto floodplains, up gentle slopes, into the quietest gullies and the boldest ridges. And wherever those plants went, fungus followed, not trailing behind, but ahead of them, whispering the way forward with chemical cues and unseen roads. The land changed, visibly and deeply. Microhabitats began to emerge. Sheltered soil supported more complex communities. Mosses and lichens began to layer, creating depth. Decayed plant matter mixed with fungal threads formed humus, a richer, darker layer of soil that could retain more water, bind more nutrients, and support thicker root systems. The biosphere thickened, but the consequences were not just local. As more plants took hold on land, more carbon dioxide was drawn from the atmosphere through photosynthesis. And as fungal activity increased below ground, more carbon was sequestered in long-lived organic compounds and mineral complexes. CO2, once thick in the atmosphere, began to fall, and Earth's thermostat responded. The planet cooled, again. This time, it did not freeze completely, not like the snowball Earth before, but the temperature dropped just enough to shape the pace and pattern of evolution. Certain life forms thrived in this cooler, more stable world. Others faded. The oceans, receiving increased input from weathered soils, became enriched with phosphorus and other nutrients, spurring periodic algal blooms. These blooms, like green tsunamis of microscopic life, drew in sunlight and exhaled oxygen, further shifting atmospheric chemistry toward conditions amenable to complex life. Oxygen too began to rise, and with it came entirely new metabolic possibilities. In a low-oxygen world, life had been slow, limited to fermentation and other inefficient pathways. But with more oxygen came aerobic respiration, a form of metabolism that unleashed far more energy per unit of food. This meant organisms could grow larger, more complex, more coordinated, and they did. Simple multicellular animals began to evolve, first as soft-bodied creatures in shallow seas, then as mobile beings capable of sensing, reacting, hunting, and building. This was the Cambrian explosion, but it didn't come from nowhere. It was lit in part by the quiet groundwork laid by fungi. On land, the changes kept building. As plants grew taller and denser, they created new microclimates. They shaded the soil, trapping moisture. They dropped their leaves, adding organic matter to the ground. They built verticality into an ecosystem that had previously been flat and textureless. Fungi adapted too. Some evolved to live entirely within plant tissues, forming endophytic relationships. Subtle alliances where fungal cells lived between plant cells, exchanging signals, repelling pathogens, enhancing drought resistance. Others specialized in decomposition. As plant life expanded, so too did plant death. Fallen stems, collapsed mats, shed leaves, all became fodder for a growing army of fungal decomposers. These fungi didn't just clear the forest floor, they returned essential nutrients to the soil. They recycled carbon. They maintained balance. And then, in one of evolution's most quiet but decisive moves, some fungi developed a taste for lignin. Lignin is what makes wood hard. It's what gives trees their strength and resilience. It's notoriously difficult to break down, so complex that few organisms even attempt it. But fungi did. White rot fungi in particular evolved enzymes capable of dismantling lignin's tangled structure, unlocking the carbon and energy stored within. This single adaptation altered the course of Earth's history. Before white rot, dead trees could persist for centuries, even millennia, building up into massive deposits of undecomposed wood. But once fungi learned to eat wood, that carbon began cycling faster. Ecosystems could support more life. Nutrients moved more quickly. Decay became more efficient. And carbon that wasn't recycled, because it was buried too deep or too quickly, became coal. Yes, the great coal beds we mine today are echoes of a time before fungi had fully mastered lignin. They are the result of partial decomposition in an age when trees were new and fungi were still learning their way through the forest. And once they learned, once decay became swift and complete, the window for coal formation quietly began to close. Another consequence, another ripple. And all the while, the planet's crust continued to shift. Mountains rose, exposing fresh rock. Rivers carried silt to new basins. Fungal spores travelled on the wind, colonizing every inch of reachable surface, always ahead of the green wave, always preparing the land for what came next. And what came next was diversity. The ecosystem born of this fungal-plant symbiosis began to layer and lace together, forming food webs. Fungi became prey for microscopic grazers, springtails, mites, early arthropods. They became hosts for bacteria. They began forming fungal-fungal relationships, even parasitizing one another in complex fungal warfare waged in soil no thicker than your fingernail. And in the fossil record, a strange new texture began to appear. Root traces, rhizomorphs, complex soil horizons, evidence not just of life, but of ecosystems knitting themselves into the ground. By the time vertebrates finally lumbered onto land, blinking in the sun, fungal networks were already woven through every clump of moss and every patch of dirt. Their spores filled the air. Their scents marked the soil. Their enzymes shaped the chemistry of everything living things touched. And that chain reaction, the one started by a thread creeping across a rock, had become unstoppable, so the chain continued. By the time amphibians crept from swamps and reptiles basked in the first sunlit clearings, the world was no longer a raw canvas but a palimpsest of cycles written and rewritten by fungus. It was there in every breath of the forest, every crumble of wood, every drop of dew sinking into leaf litter. And though never the main actor in the eyes of history, fungus was always on the crew list. Stage manager, set designer, cleanup team. Its network stitched the biosphere together, and with that stitching came resilience. When drought gripped the land, fungal threads carried moisture between plants, even across species. When one tree wilted, its neighbours, connected underground through the mycorrhizal web, might receive the chemical warning. A subtle plea passed from root to root. And though this communication had no words, it had purpose. Defences rose, stomata closed, drought was weathered together. When pathogens arrived, some fungi took on the role of shield. They produced antimicrobial compounds, natural antibiotics to protect their host plants. Others sharpened the sword, becoming pathogens themselves, keeping population booms in check, pruning the system of excess. This was not cruelty, it was balance. And balance, more than beauty or competition, is what ecosystems crave. Fungi with no hierarchy, no central brain, no ambition for dominance, became the unconscious architects of equilibrium. They transformed complexity from a liability into a strategy, and they did so across every landscape. In boreal forests, they formed vast intergenerational webs. In deserts, they created biological crusts that held sand in place, trapping moisture under blazing suns. In wetlands, they decomposed the dead while anchoring the living. And in the high altitudes of new mountain ranges, they were often the first to arrive. Lichen clinging to frost-split granite, mapping out soil where snowmelt might collect. And still they multiplied. Some fungi evolved to specialize in relationships so intimate they blurred the line between organism and organ. Endomycorrhizal fungi didn't just wrap around plant roots. They grew inside them, weaving through cell walls, never piercing membranes, but pushing right up against the edge of intimacy. There, in tiny chambers, nutrient and sugar flowed in two directions like a double heartbeat. This internal partnership allowed plants to expand into drier, harsher terrains. Grasslands emerged. Savannas widened. Trees followed rivers deeper into continents, their growth written not in bark or branch, but in the invisible scaffolding of fungal support. And the sky too responded. Plants supported by fungi drew more and more carbon from the air. This drove cooling, gradual, widespread, a planetary exhalation. As the climate shifted, new evolutionary pressures emerged. Creatures adapted, ecosystems diversified. Fungi adapted too. Some began breaking down chitin, the tough material in arthropod shells. Others found a niche in animal dung, thriving in decomposition cycles that had once been exclusively plant-based. one group evolved to hunt. Yes, hunt. Zupagomicota, for example, developed hyphal loops that could trap nematodes, tiny lassos that snapped shut when a worm brushed past, a fungus that could sense, respond, and devour. The rules were changing, the world was ripening, and fungi were always just ahead of the curve. But even paradise has its pendulum, mass extinctions. Those great punctuation marks in Earth's biological story came and went, and in every one, fungi played roles both quiet and immense. Take the Permian extinction, about 252 million years ago, the largest mass extinction in Earth's history. Over 90% of marine species and 70% of terrestrial vertebrates vanished in an ecological blink. Volcanism had opened up across what's now Siberia, unleashing floods of lava and rivers of gas. Carbon dioxide surged, temperatures spiked, oceans acidified and turned stagnant, forests collapsed, and in the fossil record something remarkable appears: the fungal spike. In layers from around the world, fossilized fungal spores suddenly outnumber pollen grains by an overwhelming margin. Entire strata are blackened with them. It was not celebration, it was response. When ecosystems die en masse, the decomposers come to feast. The fungal spike marks a moment of near-global rot, a bloom of fungi rising not to replace, but to recycle, to break down the dead, to clear the field, and once again, they prepared the ground for what came next. Each extinction brought new opportunity, each collapse reset the game, and fungi, ever adaptable, filled the voids, softened the scars, and welcomed the return of the green. And even as animals grew more complex, eyes evolved, feathers took flight, mammals found warmth in fur, fungi remained the quiet constant. Neither predator nor prey, neither plant nor beast, yet entwined with all. Some began living inside animals, helping them digest their food. Others became parasites, sometimes deadly, sometimes gentle. The line blurred between invader and symbiont. In leafcutter ants, evolution reached a new complexity. Entire fungal farms maintained in underground chambers. The ants cut leaves, not to eat, but to feed their fungi. And the fungi, in turn, provided nourishment. An insect-fungus agriculture system that predates human farming by tens of millions of years. In that partnership was a kind of foreshadowing, a hint that intelligent life, should it ever emerge, might come to depend on fungi in ways even ants could not imagine. And so it was. We too formed our bond, not just through decay and soil, but through bread and beer, through cheese and soy sauce, through penicillin and cyclosporin, through yeast and mould, through fermentation and medication, through psilocybin visions and truffle-laced indulgence. Fungi didn't just shape our planet, they shaped our culture, and still we barely understand them. Even now, the fungal kingdom remains one of the least studied branches of life. For every mushroom identified, hundreds more go unseen. Many have no fruiting body. They live in roots, in wood, in air, in skin. Some species go years between reproductive cycles. Others may have already gone extinct before we knew they were there. They are everywhere, and they are elusive. And yet, they are essential. For every tree that rises, a fungal partner steadies it. For every forest that burns, a fungal wave follows to reclaim it. For every body that falls, they are there to carry it back into the soil, into the cycle, into the waiting arms of renewal. And so the aftermath of that first fungal creep across ancient rock still echoes today. It echoes in the steam of your bread, in the spores that drift through sunbeams in your living room, in the fine threads beneath every forest trail, in the enzyme that heals your infection, in the soft decay of autumn leaves. The chain reaction never ended. It simply changed costumes. A thread that began nearly 1 billion years ago is still weaving its way through the story of life, one mycelial whisper at a time, Fungi don't conquer. They connect. They don't roar. They root. And when all else crumbles, when the forests fall, when the statues fade, when the cities return to earth, it will be fungi who come last and first. To carry, to clear, to prepare. Just as they always have. Every history has its echoes. What begins in one epoch often stirs again in another, not as repetition but as rhythm. And Earth, for all its wild variety, is a deeply rhythmic place. It breathes in eras, sighs in epochs, and every so often it pulses. A surge of life, a collapse, a rebirth, again and again and again. Fungi are not the only force to rise from silence and reshape a world, but theirs is one of the earliest and most enduring rhythms, and to understand it fully, we must step back. Not just back to their beginning, but back beyond it, to the events that set the stage for their arrival, and those that followed in their wake. Our planet, in its youth, was a hot and violent place. 4.5 billion years ago it formed in fire. Collisions, magma oceans, noxious gases. The surface boiled and buckled. For hundreds of millions of years there was no fungus to creep, no algae to bloom, no structure to soften the noise of raw geology. But even then, water was carving paths across cooling stone. And in that water, simple life, prokaryotic, ancient, persistent, began to stir. By 3.5 billion years ago, cyanobacteria were flourishing. They formed great mats in shallow seas, building stromatolites, layered structures that still survive in some corners of the modern world. They pumped oxygen as a waste product, slowly poisoning the anaerobic world they'd known. This was the first great metabolic revolution, and with it came one of the first great crises, the Great Oxygenation Event around 2.4 billion years ago. Oxygen, though essential to us now, was toxic to most early life. Its increase triggered a mass extinction, perhaps the first. Entire microbial ecosystems were wiped out. But in the ruins, new pathways opened. Respiration became more efficient. Cells grew more complex. Eukaryotes, the ancestors of fungi, plants and animals, emerged. That transition, invisible to the naked eye, rewrote the logic of life, and it laid the molecular groundwork for everything that followed. Now leap forward, past billions of microbial years, past the snowball Earths, past the slow dance of continents, to the emergence of multicellular life. It began in water, first as soft-bodied organisms, then as animals with symmetry, mouths, muscles. The Cambrian explosion, around 540 million years ago, was not an explosion in the fiery sense, but in the biological sense. Diversity bloomed, eyes evolved, shells hardened, nervous systems blinked on. The tree of life branched furiously outward, and beneath it all, in the sediment, fungi were already weaving. They're not part of the Cambrian story that gets told in museums and textbooks. The trilobites get the fame, but their precursors were likely active in coastal shallows, interacting with microbial mats, starting to inch toward the shore. And when that shore became green with plants, fungi were already waiting. This was Earth's second great greening, the first being the cyanobacterial oxygenation of the ocean. Now the land joined the biosphere. Mosses, liverworts, then vascular plants, then trees. Roots dug deep, forests rose, oxygen levels surged again, and in response, life grew larger. Insects reached wingspans the size of falcons. Amphibians colonized ponds. The Carboniferous period was an era of swamp and shadow, of dense canopies and humid ground. And the coal deposits we mine today are time capsules from that age, a time when trees died faster than fungi could decompose them. But that balance didn't last. By the end of the Carboniferous, fungi had caught up. White rot fungi, with their lignin-destroying enzymes, began recycling wood efficiently, and the carbon that once built up into coal now cycled back into the atmosphere and soil. a gentle closing of a loophole in Earth's carbon budget. Meanwhile, tectonics continued to drift continents into collisions. Mountains rose, weather patterns shifted, new habitats opened, and extinction, as ever, came in waves. Earth has endured 5 major mass extinctions. Each one cleared the board, each one made room. The Ordovician-Silurian extinction, 444 million years ago, was triggered by glaciation. Sea levels dropped, habitats vanished, life regrouped. The Devonian extinction, 372 million years ago, was slower, perhaps caused by expanding plants altering soil chemistry and triggering anoxic ocean events. Again, fungi were involved, not as destroyers but as witnesses and recyclers. Then came the big one, the Permian extinction. 252 million years ago, volcanic eruptions in what is now Siberia poured out carbon dioxide and methane, warming the planet. Oceans turned to poison. Over 90% of species disappeared. And there, again, the fungal spike. In the ash and soot, in the silence that followed, fungi bloomed. They fed on the fallen. They turned ruin into compost. And when life returned, more cautious, more reptilian, it did so on soil laced with fungal chemistry. Later came the Triassic extinction, opening the door for dinosaurs. And then, most famously, the Cretaceous event, 66 million years ago, when an asteroid's impact darkened the skies and ended the Age of Giants. In each case, fungi surged in the aftermath, not just in fossils, but in function. After the dinosaurs fell, mammals rose, angiosperms spread, forests reshuffled, fungi diversified again, tracking the changes, adapting to new hosts, new niches, new strategies. They evolved to ride beetles, mimic insects, infect frogs, decompose oil, and communicate with trees. And today, they remain in the background of every ecosystem, but always at the centre of its function. They are the memory of Earth's prior selves. And when we pull back even further, we see a pattern emerge, a cycle. Earth warms, then cools. CO2 rises, then falls. Diversity surges, then collapses, then surges again. Through it all, fungi remain. Agents of decay, pioneers of growth, architects of recovery. They remind us that life's progress is not linear. It is layered, interwoven, recursive. Each mass extinction creates space for new forms. Each collapse reshuffles the genetic deck. And the organisms best equipped for this rhythm, the decomposers, the adapters, the partners, are not the flashiest but the most flexible. Fungi are that flexibility made manifest. And in the context of climate change today, their story becomes more than ancient history. It becomes a mirror. Once again, CO2 is rising. Once again, the planet is warming. Once again, ecosystems are shifting, species are vanishing, niches are opening, and once again, fungi are responding. In permafrost, awakened by thaw, fungal species are beginning to decompose carbon frozen for millennia. In cities, moulds evolve rapidly in subway walls, gutters, air vents. In forests stressed by drought, mycorrhizal networks change allegiance, rationing resources to species most likely to survive. Fungi are adapting, but they cannot fix everything alone. We are no longer outside the system, observers of a planetary cycle. We are part of the mechanism. We are the triggering event. And perhaps, just perhaps, there is something to learn from the ones who have survived every collapse, every shift, every age of fire and frost. Fungi do not dominate. They connect. They do not rush. They persist. They do not control. They cooperate. And in that, there is a kind of wisdom, non-verbal, chemical, ancient. A way of being that doesn't strive to shape the world into one's image, but to shape oneself to the rhythm of change. It is humbling to think that the most adaptive, resilient lineage on Earth began as a network of filaments on a bare stone cliff almost 1 billion years ago. It is more humbling still to know that they are still here, beneath us, within us, among us, not as fossils, as participants, as teachers. And if we are wise, we'll listen, not with ears, but with questions. Not to what they say, but to how they live. Because the broader context is not just one of mass extinctions and recovery. It is one of relationship. Life builds complexity not through competition alone, but through collaboration. And fungi, as early as any form of life, understood this. They didn't outgrow their competitors, they joined them, and in doing so, they helped life find the edges of its possible. Science, for all its equations and instruments, often begins in stillness. A pause in the woods, a question at the edge of a microscope slide, a fossil found not with fanfare but by accident. And so it was with fungi. Not discovered exactly, they've been part of human life since before writing, but slowly re-understood, re-noticed, re-wondered at. In the deep past, fungi were not subjects of study. They were symbols, omens, mysteries. Ancient people saw mushrooms appear overnight and vanish just as quickly. They fed on the dead, thrived in darkness, grew in rings without seeds or stems. In many cultures, that strangeness marked them as sacred, or dangerous, or both. The ancient Egyptians believed mushrooms were the food of royalty, delivered by lightning, forbidden to commoners. The Aztecs used Tionanacatl, the flesh of the gods, in spiritual rituals. In Siberia, shamans ate the fly agaric to pass into other worlds. Even in medieval Europe, mushrooms carried a kind of hush, feared, respected, carefully collected at dawn by those who knew which would nourish and which would kill. But none of these traditions asked, what are fungi? That question would wait. The scientific study of fungi, mycology, did not emerge as a discipline until surprisingly late. For centuries, fungi were lumped in with plants, a sort of grey botanical footnote. After all, they grew in the ground. They didn't move. They had no fur, no feathers. Surely then they were a sort of plant. A lesser one, odd perhaps, but vegetable. It wasn't until the invention of the microscope that the first real clues began to emerge. In the 1600s, Antony van Leeuwenhoek, curious Dutch lens grinder, cloth merchant and father of microbiology, peered through a handcrafted microscope and saw for the first time the hidden world of filaments and spores. He described the mould on bread as little plants with branches and bulbs. It wasn't wrong, but it wasn't quite right either. Still, it was a start. A century later, scientists like Pier Antonio Micheli began studying fungal reproduction in earnest. Micheli observed that mould spores could produce the same kind of mould when placed on fresh substrates, a radical idea at the time. It challenged the prevailing belief in spontaneous generation, the notion that life could emerge from non-life, as if decay itself birthed mould from nothing. Micheli's work suggested otherwise, that spores begat threads, begat mould. It was a cycle, not a magic trick. His drawings of hyphae and sporangia, some of the earliest, are astonishing in their detail and remain instantly recognisable to modern mycologists. Yet still, fungi were largely misunderstood. They weren't animals, they weren't plants, they were something in between, or beyond. And in truth, they were. It wasn't until the 20th century that fungi were fully split from the plant kingdom and given their own place on the tree of life. This was not a small shift, it was a tectonic one, and it came not from mushrooms, but from molecules. With the rise of molecular biology, scientists began to compare DNA sequences across life forms, and what they found was astonishing. Fungi were not only separate from plants, they were closer genetically to animals. They breathe oxygen. They digest their food externally like an inverted gut. They use similar enzymes. And their common ancestor with animals likely swam, unlike any plant. This revelation rearranged the mental map of life. It reminded us that appearances deceive. That function is not always visible, that the networks underfoot may have more in common with our own than the leaves above. But even as science sorted the branches of the fungal family tree, the fossils remained stubbornly elusive. Fungi, after all, are soft. Most of their bodies are filaments, one cell thick. Their spores are dust. They don't fossilize easily, and when they do, they rarely announce themselves. Still, some moments of luck and clarity have pierced that veil. In the 19th century, in the coal-rich lands of Britain and the United States, fossil hunters began noticing something strange. Amid the petrified ferns and giant club mosses of the Carboniferous were these narrow branching forms. Sometimes they formed nets, sometimes they pierced wood. Some were so well preserved you could still see the cell walls. They weren't roots, they weren't worms, they were fungi. And in 1859, coincidentally the same year Darwin published On the Origin of Species, a fossil was described from Scotland. It had no leaves, no roots, no flowers, but it stood nearly 20 feet tall. Prototaxites, they called it. For decades, no one could decide what it was. A tree, a giant alga, a mushroom. It had a trunk-like form, but no rings, no vessels, no chlorophyll. It wasn't until the early 2000s that a team led by Francis Huber and Carol Houghton reanalyzed its structure and chemical makeup and confirmed. Prototaxites was fungal, a massive trunked fungus towering over a world before true forests, a saprophyte the size of a small tree, rising from ancient soil to decompose whatever life fell around it. It's hard to describe the wonder of that realization, not just that fungi could grow so large, but that they once ruled the land. Before trees, before dinosaurs, before grass or flowers or bees, fungi stood like sentinels, monoliths of decay, guiding carbon from dead matter back into the cycle. And this fossil, hidden for millions of years in silt and stone, whispered that truth to those patient enough to listen. The 21st century has brought even more tools to the hunt. Electron microscopes, mass spectrometry, isotope analysis. These techniques allow scientists to study ancient spores, chemical residues, even tiny fragments of cell wall polymers like chitin embedded in shale. In 2022, the discovery of what may be the oldest known terrestrial fungus, found in 890 million-year-old rocks from the Northwest Territories of Canada, made headlines around the world. Researchers identified branching filaments with bulbous swellings and cell walls rich in chitin. If confirmed, this would place fungi on land hundreds of millions of years before the first plants. And the scientist who led the study, Corinne Tremblay, wasn't even looking for fungi. She was studying the layers of sediment laid down during an ancient mountain-building event. The sample, thin as a credit card, was prepared for unrelated structural analysis. But under the microscope, there it was, a network of threads, clear, deliberate, branching, unmistakably biological, not an animal, not a plant, fungus. Moments like these remind us that discovery is not just about seeking, it's about seeing, about noticing what doesn't fit and holding that mystery gently, long enough for it to speak. And in the field of mycology, that's often how it works. Fungi do not lend themselves to capture. They evade easy study. Many do not grow in lab conditions. Some fruit once every decade. Others bloom only in symbiosis with a single tree, in a single patch of forest, after the right sequence of rain and heat and decay. Even today, it's estimated that we have named only a tiny fraction of fungal species, maybe 5%, maybe less. And yet we're beginning to understand just how central they are to ecosystems, to evolution, to us. The more we look, the more we see. And yet with fungi, seeing is never the whole story. Because fungi do not always appear when summoned. They are not like mammals that can be counted, or birds that can be tagged. Their bodies are more absence than presence. Threads in the dark, unfolding beneath bark, between grains of soil, within the walls of other beings. Even in modern laboratories, fungi refuse to be easily known. They grow on their terms, they vanish on theirs. This has made mycology, a field that should be central to biology, into something of a scientific underdog. Even today, most biology textbooks barely mention fungi. Universities may offer a course or two, but rarely a department. Grants are limited. Field studies go underfunded. And yet the importance of fungi has never been clearer, because everywhere we look with new tools, genetic sequencing, ecosystem modelling, even climate forecasting, fungi keep turning up at the foundation. Take the discovery of the Wood Wide Web. In the 1990s, a quiet revolution began in forest ecology. Suzanne Simard, a forest ecologist from British Columbia, conducted a series of experiments that would overturn our understanding of how trees communicate. Working in the temperate rainforests of Canada, Simard tagged trees with carbon isotopes and traced how those atoms moved, not just within a tree, but between them. The carbon wasn't drifting passively through the air or the soil. It was being transferred actively, selectively, via underground fungal networks. Fungi, it turned out, were linking trees of different species. Young seedlings were receiving carbon from old-growth elders. Dying trees were offloading resources to neighbours before fading away. And the fungal networks were not just plumbing, they were traffic controllers. They facilitated, regulated, prioritised. This was not competition. This was communication. It was community. And it changed everything. Suddenly, forests were no longer collections of individuals competing for sunlight and water. They were interconnected systems, with fungi as the hidden infrastructure. Simard's work, though initially met with skepticism, has since been replicated around the world, and it has inspired a generation of scientists to look at the forest floor not as an end, but as a doorway. Beneath your feet, in every healthy forest, there is a network more ancient than the internet, more complex than any subway map, and it is alive. Other researchers have followed these filaments into surprising territory. Fungi have been found in the ice of Antarctica, in the radioactive ruins of Chernobyl, in the walls of space stations. One species, Cladosporium sphaerospermum, even seems to grow better in space, possibly using radiation as an energy source, a kind of fungal photosynthesis for the atomic age. Mycologists have identified fungi that digest plastic, others that decompose oil slicks, others that produce biofuels more efficiently than any current crop. Some are exploring the use of fungal mycelium as a building material, strong, lightweight, biodegradable. Future buildings might be grown, not built. Self-healing, self-regenerating, breathing quietly through your walls. And then there is the frontier of medicine. Penicillin, the antibiotic that changed the course of history, came from Penicillium notatum, a mould growing in a forgotten petri dish. It was Alexander Fleming's accident, but also his attention that gave it to us. The same mould would go on to save millions of lives in the decades that followed, especially during World War II, when the mass production of penicillin became a global priority. But that was only the beginning. Cyclosporin, which makes organ transplants possible, came from a soil fungus. Statins, which lower cholesterol and prevent heart disease, came from Aspergillus. Psilocybin, the psychoactive compound in certain mushrooms, is now being investigated for treating depression, anxiety, PTSD and addiction, with promising results. In labs around the world, scientists are sequencing fungal genomes, discovering new metabolic pathways, isolating compounds that may become the medicines of tomorrow. And always, in the background, are the field mycologists. They work quietly, often alone. They hike into forests with notebooks and wax paper. They crouch beside logs, sniff spore prints, photograph gills in morning light. They collect samples that may take years to identify. They name species no one has seen before. They are the stewards of a vast invisible archive. Some are amateurs, teachers, artists, hobbyists, who know their local woods with the intimacy of cartographers. Others are university researchers with budgets and deadlines. Some are artists in lab coats. Some are mystics with microscopes. But all are connected by a single thread: the recognition that fungi are not curiosities, but foundations. And the more we listen to them, the more they seem to offer us, not just in technology or medicine, but in metaphor, in perspective. Fungi live by dissolving the boundaries between things. They blur the line between life and death, between self and other. They are collaboration made physical, cooperation made flesh. Their lives unfold not in dominance, but in mutualism, not in conquest, but in exchange. They teach us that value can lie in what is shared, not what is owned. That intelligence can be distributed, that power can be quiet, and they remind us that what is most essential is often least visible. The story of fungi is, in many ways, a story of patience, on their part and on ours. They waited billions of years for us to notice. They built the foundation of terrestrial life before we'd even grown limbs to walk on it. They shaped the soil before we gave it a name, and now, as we face a century of change, they may hold keys we're only beginning to understand. But the people who study them, those quiet seekers in forests and labs, have already begun to listen. They are not chasing cures, they are building relationships. They're asking not just what fungi can do, but what they can teach us. Not just how they grow, but why they grow the way they do. Not just what we can extract, but what we might become if we learn to live a little more like them. Connected, patient, collaborative, rooted. And in that sense, the human discovery of fungi is not just scientific, it's philosophical, it's emotional, it's ancestral. Because somewhere in our evolutionary past, we were not separate from these organisms, we were with them, inside them, beside them, partnered in digestion, decomposition, soil building. Our ancestors walked barefoot on fungal mats and inhaled spores and built fires beside decaying logs and harvested mushrooms by trial and by memory. And now, in a world that often feels fragmented, isolated and accelerated, fungi return with a different kind of lesson. One that cannot be hurried. One that asks us to kneel, to notice, to remember, and to listen, not with language, but with presence. Because fungi do not explain, they demonstrate. And in their networks we see our own possibility of healing, of resilience, of reconnection. So when we trace their story, not just through eons of evolution, but through the hands and eyes of the people who dare to wonder, we are tracing a bridge from the ancient to the now, from the unseen to the understood, from silence into voice. When you kneel down and brush away the forest litter, what you touch, what clings to your fingers, is only the surface. Beneath it lies a machinery too small for eyes, too ancient for speech, too intricate for any single metaphor. Because to understand fungi is to step into a cathedral of processes, not built of stone, but of molecules, not lit by candles, but by electrons, not echoing with choir, but with chemistry so quiet, so elemental, it feels more like weather than intent. This is the layer of deep science, the level at which fungi no longer resemble organisms but forces. Begin perhaps with their cells. Fungal cells are curious things. They don't behave like animal cells, neatly packaged and bound. Nor do they resemble plants with their rigid compartments and chloroplasts. Instead, fungi weave filaments, hyphae, that stretch and branch, forming tunnels thinner than a human hair. These hyphae grow from their tips, extending like fingertips searching through darkness. And they don't do it blindly. They sense gradients, moisture, carbon sources, minerals, and grow toward them with eerie precision. Sometimes they pause, sometimes they fuse with other hyphae, sometimes they loop back and reinforce their networks like engineers building bridges in a city only they can see. Inside the hypha, The cytoplasm flows like a gentle river. Nutrients move, organelles drift, enzymes are carried where needed. There are no centralized organs, no bossy nucleus issuing orders from above. The intelligence of a fungus is distributed, smeared across its body like memory through a song. Some fungi have septa, internal cross walls that separate segments of hyphae, but even these are porous, allowing molecules and sometimes even nuclei to pass between compartments. Other fungi, especially ancient ones, are entirely koinocytic, one long cell with many nuclei, unbroken, undivided. Imagine a brain with no neurons, a bloodstream with no heart, a thought with no skull to contain it. That is a fungal filament. And when enough filaments tangle together, they form mycelium, the body beneath the mushroom. The part that doesn't fruit, doesn't display, but lives. Networks of threads so dense and broad they can span forests, continents, perhaps eras. The largest living organism on Earth is not a whale or a redwood. It is a fungus, Armillaria ostoya, the honey fungus of Oregon. Its mycelium spreads over 2,300 acres, estimated to be over 2,000 years old, and it's mostly invisible. How does such an organism persist, grow, sense itself? The answer, in part, lies in a kind of fungal language, electrical pulses, chemical gradients, rhythmic patterns of gene expression that respond to the world not with thought, but with transformation. Fungi don't move, but they act. When they sense a competitor, they release antibiotics. When they detect a host, they produce specialized enzymes to breach its defenses. When nutrients run low, they change their metabolism entirely, shifting from sugar to protein to fat, scavenging every last usable molecule like alchemists working with scraps. This adaptability is not random. It is encoded, emergent, self-modifying. And when food truly runs out, when the world offers nothing more, they prepare for transit. They sporulate. Fungal spores are masterpieces of survival. Some are shot into the air with microscopic cannons. Others hitch rides on animals. Some drift like pollen. Others sink into soil. Many can remain dormant for years, centuries even, waiting for the right signal to germinate. And those signals, they're precise. A trace of moisture, a slight shift in pH, a molecule of glucose, a whisper from another fungus nearby. No wasted effort, no wasted time. And beneath all this, beneath the spore and the hypha and the network, is the genome. Fungal genomes are marvels of compact complexity. Many are surprisingly small, yet filled with adaptive tools. Whole suites of enzymes to digest wood, bone, keratin, cellulose, lignin, rubber, plastic. Genes that switch on only under certain conditions. Mobile elements that shuffle, mutate, experiment. Some fungi have sex, some don't. Some do both, depending on the day. Some have more than one nucleus in the same cell, cohabiting, cooperating, sometimes even competing. The rules, again, are blurry, and this genomic flexibility gives fungi an evolutionary agility unmatched by most organisms. They evolve fast, they hybridize, they adapt to chemicals, they colonize new environments with dizzying speed. And in our age, an age of synthetic biology and climate chaos, that agility matters. Because fungi are not just passive recyclers, they're active engineers. In the carbon cycle, fungi are the final act. When plants die, it is fungi that break them down. When animals fall, fungi decompose flesh, bone, hair, hooves. They release carbon dioxide back to the air. They fix nutrients in the soil. They transform waste into structure. In doing so, they influence global carbon levels. Too many fungi and decomposition outrun storage. Carbon floods the sky. Too few fungi and dead matter accumulates. Ecosystems stall, nutrients lock. Decay slows. The balance is exquisite, dynamic, atmospheric. And fungi aren't just carbon players. They shape the nitrogen cycle too. Some partner with bacteria to fix nitrogen. Others release enzymes that convert complex nitrogenous waste into forms plants can use. They influence phosphorus availability, iron uptake, sulfur oxidation, methane suppression. Some fungal metabolites feed into the weather itself, acting as cloud condensation nuclei. In boreal forests, spores released by mushrooms can influence rainfall. A mushroom becomes not just an organism, but a raindrop's midwife. And perhaps most startling of all, fungi can shape rock. Through chemical weathering, fungal acids dissolve mineral grains. They pry calcium from feldspar, phosphate from apatite. They oxidize metals. They sculpt soil. The biosphere at its root is a geochemical engine, and fungi are among its finest mechanics. They don't just live on Earth, they write it. And as we gaze beyond Earth, toward Mars, toward Europa, toward the icy moons of Saturn, fungi have become unexpected players in astrobiology. Because if life exists elsewhere, especially life that survives in radiation, in salt, in darkness, fungi may be the best analogues we have. Some researchers are studying how fungi grow under Martian conditions. Others are testing whether mycelium can form radiation-proof habitats for future astronauts. Still others ask whether ancient fungal metabolisms could be replicated with alien biochemistry, proteins that fold differently, acids that don't match ours, but processes that rhyme. And the results so far, fungi survive. On the outside of the International Space Station, spores of Aspergillus and Penicillium have endured years of vacuum, UV and cosmic rays, waking upon return to Earth as if from dreamless sleep. On the Moon, in the lunar regolith returned by Apollo, researchers found no fungal life, but the possibility lingers. Because fungi don't need sunlight, only chemistry. If the universe has a shadow life, a deep, slow, patient one, it may not look like us. It may look like thread. To speak of fungi is to speak in networks, not just physically, but philosophically. Their very way of being defies the straight line. They don't build hierarchies, they build meshes. They don't follow linear commands. They respond to gradients, to signals, to whisper trails of change. And more, this pattern, this distributed decentralized adaptability, is sounding familiar. Not in biology, in computation. Scientists and systems theorists have begun to look at mycelium as a kind of natural intelligence. Not because it thinks, not because it calculates, but because it adapts. And adaptation, it turns out, is a kind of cognition. In laboratories across the world, researchers have begun growing fungal networks on petri dishes shaped like mazes, cities, transit maps. They place nutrients at key junctions and observe how the mycelium navigates. And time and again, it finds the most efficient path. Not always the fastest, not always the simplest, but the resilient one, the one that can reroute if broken, that can adapt if interrupted. In one famous experiment, Physarum polycephalum, a slime mould, fungal in spirit if not in classification, was placed on a map of Tokyo with oat flakes marking train stations. Within hours, the network of filaments it created mirrored the actual Tokyo metro map with striking efficiency. No blueprint, no algorithm, just hunger, memory, and flow. This isn't intelligence in the human sense. There are no thoughts, no decisions. But it is a form of processing. Environmental input, internal change, outward structure. And that has inspired a growing field of research, biocomputation. What if, instead of building machines that mimic brains, we built systems that mimic mycelium? Distributed, regenerative, embedded in the substrate of the world itself. Already, experiments are underway using fungal networks as analog computers, capable of solving logic problems, optimizing pathways, even reacting to electric pulses in ways that resemble primitive learning. It's still early. The fungi aren't running operating systems, but they respond. And that responsiveness hints at something deeper. an embodied intelligence, not seated in a skull, but spread across contact, shaped by interaction, memory as movement, logic as growth. And this touches something even broader, because if fungi show us anything, it's that intelligence doesn't have to look like us. It doesn't even have to think like us. It just has to work. And fungi work. They work in forests. They work in ecosystems. They work in partnership. Consider the lichen. Half fungus, half photosynthetic partner. Algae or cyanobacteria living as one. Not a merger, not a parasite. A mutualism so tight it forms a third being. The lichen isn't just fungus plus algae. It's something new. A compound life. In recent years, even this simple story has grown more complex. Researchers discovered that many lichens also contain yeast, single-celled fungi living on their surface. Not contaminants, not invaders, partners, perhaps essential, perhaps regulatory, perhaps communicative. A lichen, then, is not 2 species but 3, or more. A tiny microbiome walking the edge of survival. And what is it doing? Decomposing rock, seeding soil, anchoring the barest edge of life. This is what intelligence looks like when it has no eyes, no language, no hunger for dominance. It looks like weathering, like patience, like systems learning how to persist by sharing space. And if we take that seriously, if we see fungal life not just as function, but as form, then we begin to glimpse new directions. Directions in engineering, directions in sustainability, directions in how we relate to Earth itself. Already mycelium is being used to grow furniture, packaging, insulation, bricks, textiles, biodegradable materials with low energy cost, high durability, and zero waste. The organism lives, builds the form, then dies, or sleeps, leaving behind a structure grown from intention and decay. Some researchers imagine entire buildings grown this way, not built in factories but cultivated, reactive to moisture, self-healing, homegrown from local strains of fungi adapted to the climate. A fungus brick might sprout insulation in winter, exhale moisture in summer, expand or contract with the needs of its environment. Other visions are even stranger. Fungal clothing that responds to sweat, smell, or heat. Fungal drones that decompose after delivering emergency aid. Fungal coffins that break down bodies without chemical embalming, returning nutrients to soil with reverence. And still others ask, could we live with fungi, not just use them? Could a home include active mycelium, regulating its own microclimate, digesting household waste, emitting subtle scents based on air quality? Could our urban architecture stop resisting nature and begin hosting it, building it into its very bones? It's not a fantasy. The technology exists. The challenge is not the science, it's the shift in mindset. And here, again, fungi might be our teachers, because they do not fight the systems they're part of. They respond to them, reshape them, soften them. They're adaptive architecture, living feedback. And feedback is the key to survival. As Earth's climate shifts, as carbon rises, as heat deepens, as ecosystems tip, fungi are already changing. In the Arctic, as permafrost thaws, dormant fungi wake. They accelerate decomposition, releasing ancient carbon and methane. The process feeds itself. In temperate forests, drought stresses trees. Stressed trees offer less sugar to their fungal partners. Some fungi switch hosts, some die. Others mutate, shifting their enzyme profiles to tolerate dryness. In cities, fungi evolve rapidly. They grow in gutters, in walls, in air ducts, in shoes. Some become more allergenic, others less. Some acquire resistance to antifungals, shaping the medical future in real time. In all this, fungi are not the villains. They are the mirror. They show us the rate of change, the direction of response, the fragility of the balance. And they offer us a tool set, not just in carbon cycling or waste reduction or regenerative design, but in thinking. Fungal thinking. networks, not nodes, exchange, not extraction, persistence, not permanence, learning not as memorization, but as mutation, rooted in contact, grounded in response. And in that shift lies a possibility, not of domination, not of returning to some imagined balance, but of aligning ourselves with a system older and more adaptable than any we've built. Fungi have survived 5 mass extinctions. They may help us through the 6th, not as saviours, but as models, and perhaps one day as neighbours. Time does not heal all wounds, but it does soften them, erode their edges, bury them beneath layers of sediment, silence, and new life. After each extinction, each planetary pause, there is a space where life holds its breath, a lull in the music, a hush in the forest that hasn't grown back yet. And into that space something must step first. Often it is fungi. After the Permian extinction, the worst the world has ever seen, over 90% of marine species and most land vertebrates vanished. Ecosystems collapsed. Forests fell. Coral reefs dissolved. There were no flowers yet, no grasses, no mammals, no birds, and for a time, very little moved at all. But the fungi moved. They moved not with legs or wings, but with hunger and filament. The fossil record tells us that after the Great Dying, as it's sometimes called, fungal spores surged. Scientists call it the fungal spike. Across what is now Russia, China, South Africa, India, spore layers piled up in the rock, black flecks of life amid layers of ash and absence. This was not a celebration. It was cleanup. Fungi do not need sunlight. They do not need companionship. They need moisture, carbon, time. And in the smouldering wreckage of that ancient world, there was plenty to digest. They turned death into matter, matter into soil, and soil into potential. This is the beginning of recovery. Not the return of life as it was, but the beginning of something new, something smaller, slower, simpler. In the millions of years that followed the Permian extinction, early conifers crept back across scorched land. Ferns unfurled again beside blackened riverbanks. And the first of the survivors, small reptiles, hardy amphibians, followed. And underground, the fungal networks rebuilt themselves. They had not vanished. They had fractured, withdrawn, waited. Now they returned to form new partnerships. With the first plants to regrow, they re-established mycorrhizal webs, channels for water, phosphorus, nitrogen, and sugar. Hyphae snaked through ash-laden soils, finding roots, anchoring life where nothing else could thrive. Some fungi adapted to feed on new materials, charred wood, scorched pollen, bones left unburied. Others specialized in stabilizing dust and debris, forming crust that held moisture, shaded young seedlings, made room for moss. They were not quick about it. Recovery after the Permian extinction took tens of millions of years. Entire geological periods passed before biodiversity even began to resemble what came before. Coral reefs didn't reappear for another 14 million years. Complex forests took longer still. But fungi were there the whole time, and in the fossil record we see this quiet persistence, thin filaments in petrified root zones, spores in lakebed sediment, traces of fungal enzymes in the molecular scars of ancient peat. This was not a sprint. It was a soil building, a reweaving of the biosphere's threads, and it happened again after every extinction. After the Ordovician glaciation, fungi colonized newly exposed rock and seeded soil for early land plants. After the Devonian die-off, when the first forests fell, they recycled the lignin that nothing else could. After the Triassic collapse, they fed on dead gymnosperms and cleared paths for the rise of dinosaurs. And after the asteroid hit at the end of the Cretaceous, blocking the sun, boiling forests, collapsing food chains, fungi once again bloomed in the darkness, not in joy, in service. The ecosystem, broken and burned, was once again turned under and composted by the kingdom that never forgets how to begin again. And always, after the composting comes the bloom. Each extinction was followed not by stasis, but by diversification. In the wake of silence, evolution sang again. Different melodies, different players, same rhythm. After the Permian, mammals and archosaurs found space. After the Cretaceous, birds and flowers filled the sky and ground. And through it all, fungi adjusted. They partnered with new plants, infected new hosts, adapted to new climates, created new niches. They were the threads beneath every recovery, but the consequences of their role went deeper than biology. They reshaped the carbon cycle. By decomposing the dead and cycling nutrients, fungi determined how much carbon stayed in the soil and how much returned to the air. In wetter eras, when decomposition was slower, more carbon stayed buried, leading to global cooling. In drier times, fungi worked faster, sending more carbon skyward, fueling warming. This feedback loop, subtle but profound, helped steer the climate, and climate in turn shaped evolution. Cold ages favoured furry mammals, warm eras birthed jungles, ice advanced, life retreated, and fungi, ever present, adjusted their pace to match. So the long-term consequence of fungal resilience is not just survival, it is guidance. They are part of Earth's thermostat, its recycling crew, its restoration team. And now, in this present moment, this strange and uncertain Anthropocene, they are still doing what they have always done. They decompose, they redistribute, they repair. In forests ravaged by wildfire, fungi are among the first to return, sprouting fruiting bodies from blackened logs, sending hyphae through scorched earth, building scaffolds for new root systems. In cities they digest pollution, filter wastewater, grow in gutters and cracks and walls, holding crumbling infrastructure together with invisible glue. In agriculture they regulate soil health, resist pathogens, help crops survive drought, heat and disease. And in our bodies, they remain essential companions, regulating digestion, producing compounds, balancing microbes, sometimes making us sick, but more often keeping us well. We are not post-fungal creatures. We are part of their recovery too, and perhaps, if we pay attention, we can help them help us. Because there are places, too many, where the networks are fraying. Deforestation breaks the mycelial grid. Monoculture farming strips the soil of fungal partners. Pesticides, fungicides, pollution all sever the threads, and when the networks break, so does resilience. Plants grow weaker, soils erode, carbon is lost, the system grows brittle. But the good news, the patient, hopeful, fungal news, is that healing is possible. Fungi do not need perfection. They need opportunity. Replant a native tree. The spores will find it. Leave a log to rot. The mushrooms will return. Let the soil rest. The threads will regrow. And with them, the balance. Not instantly, not all at once, but surely. Because that is the lesson of long-term recovery. Life does not bounce back. It grows back. Thread by thread, root by root, year by year. Not in the image of the past, but in a new form, informed by the wounds, shaped by the scars, wiser, slower, more complex. Fungi teach us that recovery is not a return. It is a remaking. It is messy, uneven, full of failure and breakthrough. It is slow, gloriously, defiantly slow. And in a world that moves too fast, that forgets too quickly, that demands results before systems are ready, fungi remind us, some things cannot be rushed. A forest takes decades, a partnership takes centuries, a soil, once stripped, may need generations to regain its voice, but it can, and it does. Because life, when given a sliver of stability, will find a way. Not always the way we expect, but a way. A path through ash, a bloom after frost, a hyphae in the dark, And from that single thread, if we let it stretch and bend and braid, recovery begins again. Some stories don't end. They fade. They quiet down like a campfire collapsing into coals. And yet, even in that hush, they continue, underground, unspoken, alive. The story of fungi is one of those. Not a climax, but a continuum. A whisper that began before words and will likely outlast us. So what then can we take from it? What does the long, slow unfolding of this kingdom, the rise of its filaments, the fall of its forests, the way it weaves and waits, have to do with us now, in this hour, in this age? Perhaps more than we'd like to admit. We live in a world obsessed with growth, with speed, with competition, taller buildings, faster networks, louder voices, harder edges. The more visible, the more valuable. But fungi have never worked that way. They grow sideways, downward, inward. They connect rather than climb. They recycle rather than replace. They persist without spectacle, and in their shadowed silence there is a model, a way of being that doesn't dominate its environment but collaborates with it, a way of knowing that isn't linear but radial, that spirals through context, that listens, adjusts, responds. And this might be what we need most right now, because if you listen closely, truly listen, you can feel the earth changing, not just in data, but in pulse. Weather is strange, seasons blur, forests flicker, oceans warm, insects vanish without farewells. Coral goes pale like memory lost in real time, and through it all we search for solutions. We engineer, legislate, argue, optimize. But rarely do we pause to ask, what is the system beneath the system? What holds resilience in place? And often the answer is fungi. They are not a footnote to ecosystems. They are ecosystems. They bind the soil, they feed the trees, they digest the waste, they carry the messages, they remember the shape of forests long gone and carry those instructions in spore and strand. They are Earth's feedback loops, its resilience matrix, its long-term memory. And we are breaking them with pavement, with poison, with forgetfulness. When we fragment forests, we don't just lose trees, we sever mycelial networks that may have been building for centuries. Networks that once linked elder trees to saplings, soil to seed, beetle to bark. When we sterilize farms, we don't just kill pests, we erase partnerships, fungi that once extended root systems, filtered heavy metals, buffered droughts. When we ignore fungi, we misunderstand how life actually works, not in silos, not in winners, but in webs. And yet, there is still time. Fungi are patient. They have waited through extinctions. They have rebuilt after collapse. They do not ask for credit, only space, only time, only not to be forgotten. And in return, they offer us a mirror. Because we too are networks. We too are shaped by relationship, by invisible threads, by exchanges too subtle to name. The mycorrhizal threads beneath a forest floor are not so different from the bonds between neighbours, friends, families. The way we share stories, the way we build trust, the way we nourish one another through dark seasons. The internet too was built in fungal logic, decentralised, redundant, resilient. And yet we so often forget the nature of networks. We rush to individualism. We mistake visibility for value. We uproot what we don't understand. Fungi remind us the most vital structures are often unseen. The most meaningful change happens underground, not as revolution, but as decomposition, as transformation, as the slow turning of what was into what might be. This is not a metaphor, it's a strategy. In a time of climate collapse, rapid extinction, social unravelling, perhaps our most urgent work is not innovation, but digestion. To compost what no longer serves, to break down systems that extract, exploit, exhaust, to turn dead paradigms into nourishment. Fungi show us how. They don't fight what's dying, they accept it, They move in, they break it down, and from it they grow. It's not destruction, it's alchemy. And that's not to say the process is easy. Fungal growth is often slow, uncertain. It meets resistance, it gets pruned, but it continues, and so must we. Because the parallels don't stop at metaphor. The future of food, fungi. The future of medicine, fungi. The future of packaging, construction, planetary colonization, fungi. And deeper still, the future of thought. Already the edges of neuroscience, ecology, philosophy and computation are touching on fungal logic. What does it mean to think like a network? To build intelligence without centralization? To distribute memory across connection rather than storage? Fungi may hold keys not just to sustainability, but to entirely new ways of perceiving agency, consciousness, even time. They do not age like we do. They do not hurry. They do not fear stillness. What we call decay, they call beginning. And perhaps that is the deepest lesson. We fear death. They feed on it. We fear endings. They build from them. To fungi, rot is not a failure. It is the moment before rebirth. In that truth, there is something radical, something hopeful. Because it means that our moment, this era of unravelling, of crisis, of systems fraying, is not beyond repair. It is ripe. It is soft. It is ready. Ready for threads to weave through it. Ready for partnerships to take root. Ready for a slower, deeper kind of intelligence to rise. Not from above, but from below. From the ground. From the mycelium. From the parts of ourselves we've neglected, suppressed, or forgotten. Fungi are not just reminders of Earth's past. They are participants in its future. And so are we. If we listen, if we kneel, if we remember that to be human is not to stand apart, but to stand within. Within the soil, within the thread, within the rhythm. And as the light fades and the story slows, that's perhaps the final softest echo. Not everything needs to grow. Not everything needs to shine. Some things, maybe the most important things, just need to connect. And that, quietly, is enough. Hello again, sleepy traveller, and thank you for wandering this long mycelial path with me. We began tonight on Bear Rock nearly 1 billion years ago, and followed the quiet threads of fungus all the way to the present moment. Right here, wherever you are, whenever you are, breathing softly in the glow of a screen. So before you slip fully into dreams, let me ask one last time, where are you listening from, and what hour has the clock offered you now? Strange, isn't it? How a story that started in deep time can fold itself into a single heartbeat of your evening. If you found comfort in these winding reflections, if the science soothed you, or the imagery lulled you a little closer to rest, I'd be ever so grateful if you whispered a like or tapped subscribe. It helps this quiet corner of curiosity reach other minds in need of gentle wonder. And should you feel called to share, leave a note below, a favourite fungal fact, a question, perhaps just a greeting from your patch of earth. I read them, and each one feels like another filament joining the network. Next time, we'll drift into another pocket of planetary mystery, maybe the secret language of whale songs, or the mathematics hidden in snowflakes, wherever the night air nudges us. But for now, let the spores of thought settle, let the darkness thicken like rich soil, and let your own internal rhythms take over. Rest easy, friend of the forest floor. The fungi are on watch.